So it's my pleasure to introduce our speaker for today, Herdeline, and you go by Diggs, right?
Yeah.
Yeah, Diggs is actually grew up in the Philippines in Valenzuela. Is that how you pronounce it?
Venezuela. It's usually
Venezuela?
confused with Venezuela, but the country, but
Yeah,
yeah, it's Valenzuela city.
yes. And actually got her Bachelor of Science degree in chemistry, summa cum laude, from the University of Philippines in Diliman. And then she went on to Johns Hopkins and studied chemistry there. And then after that, just most recently was at the Weiss Institute for Biologically Inspired Engineering and the John Paulson School of Engineering at Harvard. So Diggs just joined UCI last fall, and she's doing some really interesting research that overlaps with many areas on campus, including, of course, the Beckman Laser Institute, because we deal with light so much here. And the title of her talk is Hierarchical Strategies Towards Biointerfacing with Soft Optoelectronic Materials. So Diggs, thank you very much. And we're all anxious to hear what you're going to talk about.
All right. Well, thank you so much, Tom, and thank you for this opportunity to give a talk to the Beckman Laser Institute community. Again, as I was saying earlier, I'm really appreciative of the generosity of people at BLI for helping our group set up. But today I'm really excited to share with you some of, let me actually just put this in full screen and make sure that everyone's seeing the full screen of my, okay, good, yeah. Yeah, so I'm just very excited to share with you some of my previous work and then also maybe give a little bit of snippet about what we're currently doing in the lab. But before I start with my talk, I want to share with you this slide and acknowledge my research group. As Tom mentioned, we just started last year, officially started in October of last year. So we are a young group. And it's been a challenge, of course, starting during this time. But I want to highlight the students and the group members that have done a lot of amazing things in the lab. So I'm just very proud of this group of students. I believe some of them are in this call. My graduate students, Sue and Yu Yao, have been really doing a great job leading the group. And as I said, at the end of this talk, I will be giving a small update about what we are currently doing. But for the most part, I will be talking about some of my previous work, highlighting some of my work on optoelectronic materials and building biohybrid platforms. So that's why I want to acknowledge the previous groups as well that I've been part of. So Professor Kit Parker's Disease Biophysics group at Harvard, and Professor J.D. Tovar's group, where I did a lot of the optoelectronic peptides work and received a lot of training in optoelectronic chemistries in that lab. And I was co-advised as well in Professor Haitran Mao's group, Johns Hopkins, which really opened a lot of things for me in terms of biomaterials. So with that, I just want to start this talk by giving a little bit of introduction about biointerfacing, since I will be talking a lot about that throughout this talk. And when we say biointerfacing in the context of tissue engineering, we have to consider the cell sources, the cells that we use, and the tissue architecture that they form. And of course, the materials that are interfaced with them, the materials that can be used to modulate the behavior of the living systems. And my research interest really lies on this, developing biomaterials, utilizing different chemistries, and also controlling their properties so that we can understand better about the living systems that are interfaced with them. And so for biomaterials, to date, there has been a lot of beautiful work that has already been done in order to understand how can we properly deliver cues such as chemical, mechanical, or topographical that's important and that's similar to what's found in the native tissue environment. But I would say an emerging trend and perhaps a current gap in the field of biomaterials is introducing certain functionalities that are not necessarily found in nature, but can be used in order to modulate specific cells, modulate the behavior of specific cells. So, for example, controllable delivery of electrical cues, which will be useful for excitable cells such as neurons and cardiomyocytes, and thinking about how these cells are central to a lot of debilitating diseases. And also developing some photonically active or optically active materials, which we can then use those types of functionalities for sensing applications. Now, for throughout this talk, I will be primarily talking about materials with biological macromolecules as building blocks. So primarily, I will be talking about peptides and proteins. And from a material standpoint, they're very interesting because they form these hierarchically ordered structures wherein there's a lot of dependence on the higher order structures, and also the macro scale properties on the peptide sequences, so the primary sequence, and also the intermolecular and intramolecular interactions that predominantly is governed by hydrogen bonding. So hydrogen bonding that can control the dihedral angles, resulting in different structural motifs from the fibrils to the fibers that they can form, and eventually allowing us to control for biointerfacing how they interact with cells and tissues. So, speaking of the hierarchical ordering, this has been a motivation and an inspiration for a lot of engineering design for synthetic materials. Is that can we actually utilize these hierarchically ordered structures that's found in nature in order to impart specific function into different types of synthetic materials, such as what we see in the self-assembly of peptides, for example? And for today's talk, the aspect of biomimicry that I will be focusing on is how we can use those templated assembly that is dictated by peptides in order to really organize the assembly of pi electron systems under biologically relevant environments. So types of materials that can be optoelectronically active under physiologically relevant environments. And for the majority of this talk, I will be talking about a symmetric type of pi conjugated peptide wherein the periphery is comprised of peptide units, peptide moieties, and at the core is a pi electron system. And depending on the interactions, say the hydrogen bonding and the pi-pi interactions, depending on the environment that they are subjected to, they can form a one-dimensional assembly, such as in the TEM structures or TEM image that is shown on the right. And also at higher concentrations, they can also form hydrogels. And depending on the processing, in this case, it was shear flow assembled. So we can impart some alignment that is also important for different types of tissues. So you can think about these systems once they are assembled as a biological analog of an electrical wire, because it is the peptide that can be considered as generally electrically insulating for certain sequences. It is the peptides on the periphery that is interacting with the aqueous environment. And at the core, you have these bioelectron system that can be photo electronically active or electronically active, depending on their ordering, and based on how they facilitate the mobility of carriers within these conduits, carriers that can be externally injected or can be photo induced. So I also want to note that the assembly, as I mentioned earlier, can be controlled by the microenvironment. And the reason I say that is because, let's say we have a sequence that has a lot of aspartic acids or glutamic acids. So at high pH, for example, there's a lot of negative charges due to the carboxylates that lead to coulombic repulsion. So the assembly is not facilitated. But then at lower pH, wherein these carboxylates will be protonated, the hydrogen bonding can be facilitated. And of course, the same type of logic can be applied if we control the ionic strength of the environment. So then the next question would be, these are very interesting from the fundamental standpoint, but where would they be relevant and what can we use them for? So I've been talking about biointerfacing, and that's one of the applications that we want to move forward to. And particularly thinking about developing optoelectronic materials that can be merged with biology. So when we say delivery of electrical cues, for example, the first thing that comes to mind when we think about biology is electrically stimulating electrosensitive cells. And typically how it's done in vitro is that we use a physical electrode. So in this case, I'm showing a carbon rod electrode, and that can control different types of behavior, say pace the contractility of cardiac cells. And even more recently, there's been a revolutionary finding about how electrical conditioning coupled with mechanical conditioning from stem cell derived cardiomyocytes can lead to better maturation towards more adult-like cardiomyocytes. But then if we think about using a physical electrode for long-term studies, especially for stem cells, which require really long-term cultures, that presents a high risk for infection, for example. And also there's really not a high spatiotemporal control over the application of electric fields. So there's been a lot of advances in terms of materials that can achieve materials that are interfaced with biological systems that can achieve these high spatiotemporal resolutions for not just stimulating but also recording electrical responses. But many of the examples that have been shown in the literature that are successful are actually inorganic in nature. So there is a disconnect there in terms of the biocompatibility aspect for long-term studies. And then, of course, thinking about other ways to stimulate, we can utilize optogenetics, which is the state of the art right now in terms of stimulating the action potentials of excitable cells using light. And this is a very revolutionary technique, of course, but then uh, we have to keep in mind that this requires a detailed protocol of lentiviral transfection, which also presents some species limitations in terms of long-term applications. So with that, the question that we can then ask is, these optoelectronically active materials that I've shown a couple of slides ago, can we actually use them for what we call as bioelectronic applications? So overall, merging these materials with optoelectronic functionalities and using them, interfacing them with living systems so that we can control certain behaviors. And so these bioelectron systems that I'm mentioning are primarily conjugated polymers. So some of the structures are shown right here, and many of them, perhaps you're familiar with the application of many of these polymers, like in light emitting diodes or in flexible electronics, or they've been used as components of for healthcare monitoring. But some of the advantages that they present over some of the approaches that I mentioned earlier is that actually using organic materials provides a soft and flexible network that allows for better interaction with the ions in the physiological environment. And we have to keep in mind that if we think about electrical currents in the physiological setting, it's actually the ions that are the main currency, or the ionic flux, which is the main currency for physiological environments. And then since these are organic materials, it's much more easily functionalizable with bioadhesive groups that will improve the biointerfacing aspect. And then a lot of the organic materials are also oxide-free interfaces, unlike their inorganic counterparts. So that's already an advantage for cell viability. And then in certain cases, there are organic materials that can generate currents at the interface upon optical excitation, and therefore providing that advantage or the possibility of shining light into these organic systems and establishing transient fields that could then be felt by the cells or tissues at the interface. So we want to we want to explore um, this question is and this idea of using these types of materials for biointerfacing. However, from the design perspective, there are a couple of challenges that we have to consider. And for the next few slides, I will be talking about some of the design exploration, design parameters that I have explored in the past, and what we've learned from that, and how we continue to use those design principles in order to create newer versions and newer generations of functional or optoelectronically functional organic materials. So first of all, for the conductive polymer, normally they are processable under organic solvents. But if we want to biointerface them, they have to be processable under aqueous conditions. So the approach here is, instead of using the polymer equivalent, perhaps use the oligomeric equivalent, um, and then utilize the peptides in order to assemble them. For example, what's shown here in the schematic is a 1D structure, wherein it's not just through bond conjugation, but there's also through space conjugation. But if we are relying on these types of non-covalent interaction for the conjugation, which is responsible for the mobility for the most part, we have to make sure that, one, we can control the assembly order, and two, that the electrical stimulation or the optical sensing properties that we want to achieve can be harnessed within the spatial temporal resolution that we want, and also within the size regime that is relevant for the applications. So then the first thing that I would like to share with everyone is that when we want to interface these systems, of course, we have to think about the different hierarchical levels, coming from the molecule all the way to integrating them, and really thinking about the aspects of the biointerfacing. So on the peptide level and designing the molecules, there's a lot of design parameters that we can consider. But what I want to share with you, firstly, is this study wherein we explored the design parameter space for just looking at simple substitutions, or you can think about this as point mutations at um, specific positions on the peptide, and doing a systematic change in the size and hydrophobicity of a model system. In this case, I'm using a quaterthiophene core with peptides at the periphery as a model system, because quaterthiophene is a known p-type organic semiconductor. Hence, it has already known to be applied in different devices in itself, and so it would be a good model system. So with these systematic changes in the amino acid adjacent to the pi electron core, we've seen significant changes and orders of magnitude changes in the rheological properties. And even as dry films, looking at the sheet resistance, there's also orders of magnitude differences that we can see, even though it's just the same template and simple substitutions of amino acids that are adjacent to the pi electron core. But mainly, um, it is also interesting, and it's also important to point out that all of these peptides that we are looking at in this family, all of them do form assemblies, and all of them do form one-dimensional nanostructures, with different nanostructure thicknesses. So it geometry wise, there's not that much difference in the nanoscale morphologies that they form. What's much more interesting is that we hypothesized in here that those substitutions at the molecular level can possibly increase what's happening in the stacking, and so we can then think that as we increase the amino acid size and hydrophobicity in between the monomers, since they are growing in a one-dimensional manner, that can increase the packing distance. And of course, this would have an effect on the photophysical properties, because then the through space conjugation and the excitonic coupling between the pi units will be affected as the distance is systematically changed. So that's indeed what we saw from the photophysical investigations. So just looking at the absorbance we can see that in comparison with this control, the basic at under basic pH, wherein they are not aggregating, and we can consider that these molecules exist in a monomer state, we can see that those with smaller amino acid are the farthest, the maximum absorption are farthest away or most blue shifted from the basic monomer state. So essentially, as the amino acid size increases, then it becomes, the absorption profile becomes more and more like the monomer state. And this is consistent with what we saw in the photoluminescence. So you can look at the glycine and alanine, which is the smallest amino acid, and they are very much quenched and redshifted, which is indicative of an aggregated state for the pi electron system. And the circular dichroism also supports this data and the hypothesis that we are thinking. Um, because you can see that the bisignate signal within the visible region, or within the absorption region, is most intense for also the smallest amino acid, where we think they are really close together. So excitation coupling is much more facilitated, and therefore this bisignate signal, as a consequence of being in a chiral environment or in a beta sheet-like structure, is much more enhanced for these small amino acids. And then we collaborated with Professor Howard Katz's group, also at Johns Hopkins, so that we can use these peptides as a semiconducting layer of field effect in a field effect transistor device. So for those who are not interested, essentially, in an FET geometry, you have a source and drain electrode that spans in the semiconductor, and then there's a gate layer that's separated from the semiconductor by a dielectric layer, and then there's a third electrode that is in touch with the gate, which basically shapes the current that we can see at the drain. So then in this case, we use the quarter-thiophene peptide nanostructures at the semiconducting layer, and then the gate and dielectric is just a silicon dioxide. And first, we use an electronically silent control just to see whether we can get any signals from there, but we didn't. So that's just to make sure that all of the electronic signals that we are getting are really from the quarter-thiophene stacking, which is what we did, what we saw. And so going back to the amino acid variation studies, so we saw those changes in the photophysical properties. And when we use them as semiconducting layers in an FET, where we can measure the whole mobility, we saw that there is also a systematic trend whereby as you increase the amino acid, the amino acid size and hydrophobicity, and as the packing distance changes, there's also a corresponding decrease in the whole mobility, which is essentially a parameter of the conductivity at the nanostructural level. You can think about it that way. So therefore, both of these studies show us that amino acid size is definitely a design parameter, a straightforward and simple design parameter that allows us to systematically change these properties. So by simply in this case, by simply increasing the side chain size, we were able to decrease controllably the carrier mobility that we saw. So these are just some examples of the studies, and and one example of a design parameter that can be varied. But from these and the other things that we learned about the design of these symmetrically assembled pi conjugated peptides, we wanted to take a step further and ask the question whether the peptides in the periphery can actually be used to scaffold multiple chromophores within a nanostructure, more so that we can mimic what's happening in photosynthetic pigments, whereby there are multiple pigments that their spatial organization is controlled by proteins, and the spatial organization of these pigments is very much important towards the efficiency of energy funneling. So keeping in mind the biointerfacing applications, the more important question is, can we control the photoinduced energy funneling within these peptide nanoassemblies under completely aqueous environments? So here the model chromophores are, we use quarter thiophene as an acceptor, and the donor, energy donor is OPV3. First, because of the size matching, and second is because the photoluminescence spectra of OPV3 overlaps very well with the quarter thiophene. So from that criteria itself, we can expect that there's resonance energy transfer that can occur, or FRET, maybe more familiar as FRET to other people. But at the same time, this donor system can facilitate exciton migration. And so we wanted to see whether we can actually combine these two within one nanostructure and then control and or facilitate both the energy migration and the resonance energy transfer that is occurring. So when we look at the photoluminescence, we definitely saw that from the 100% assembled donor, which is the OPV3, even just at 1 mole% of the acceptor, we saw that the peak area quenched down to 46%. So this is in acidic conditions where they are where they are completely assembled. And ideally, because we are quickly adding the acid, we are essentially kinetically trapping the structures in such a way that the donor is doped within the nanostructure, either as a single unit or perhaps as a co-assembled unit. These are the possible structure contributors to the spectra that we are seeing. And we know that this is the case because when I did the control the experiment, wherein I separately assembled the donor and acceptor units and then combined them in one solution, we saw that the change in the peak area upon the addition of 1 mole% of the acceptor, which is quarter thiophene OT4, the peak area only quenched down to 81%. So definitely when they are co-assembled, we saw much more higher efficiency of energy transport, which is also supported by the time resolved photoluminescence or lifetime experiments that I gathered from these set of solutions. So definitely, this is a good demonstration that we can indeed facilitate energy transport for under completely aqueous environments for these systems. Wherein before this demonstration, most of these systems are they can be used as PN junctions for different devices, but mostly as films and under dry environments. So then we wanted to expand further because our biomimicry example is for photosynthetic pigments, which have or photosynthetic systems which have a lot of pigments. So then we wanted to expand further and ask the question of, can we actually have more than two chromophores and try to see a controlled funneling from 2 to 3 chromophore system. So in this case, we know from our previous work and also supported by the literature that the OPV3 and OT4 can facilitate or can act as donor acceptor pairs for resonance energy transfer and also exciton migration. But then based from literature, we are also adding a third component, a third chromophore, naphthalene diamine, which has been previously shown as an electron transfer acceptor for quarter thiophene. So um, so we don't just want to extend the number of chromophores, but in this study, we further asked the question of, can we in one pot or one system control the rate of pH change? So as I mentioned earlier, it's mostly kinetic trapping that's happening when we add the acid, and it's very fast drop in pH. But if we slow down the drop in pH using a gluconodelta lactone, which hydrolyzes slowly over time and therefore can change the pH slowly within the solutions, and design each of the monomer in such a way that they will have different pKa's, so that we can achieve a stepwise assembly and achieve a self-sorted co-assembly. rather than having a fully co-assembled system. Can we use this strategy in order to modulate the types of energy transport processes? And what I mean by that is that from the results also that I showed earlier, in a completely co-assembled environment, we can expect that the energy migration is much more facilitated. And then in this case, once let's say the excitons migrate from the OPV3 to the OT4, the OT4 acts as an electron donor to the NDI. So it's mostly a sequential, the sequential energy transport processes can be much more facilitated in the co-assembled system. But in the self-sorted system, the resonance energy transfer is not as highly, well, it can occur if they are near each other, if each of these bundles are near each other. But then the exciton migration or the energy transport, similar to this, is not much facilitated. So first of all, in order to answer those questions, we wanted to prove that by using GDL, slow hydrolyzing lactone, that we can definitely achieve the self-sorted system. So we did this study in collaboration with Dave Adams' group and Louis Serpel's group at University of Liverpool and University of Sussex, respectively. So when we did NMR studies, we saw that there's a decrease, a sequential decrease in pH from the different components, which proves that there's sequential assembly. Of course, as they aggregate, the NMR peaks decrease, and uh, and then we can confirm that the gels can be formed, hydrogels can be formed from this system way after the assembly of both of the components. But really, the data that showed us that they can definitely self-sort is from Louis Serpel's group, wherein they did fiber XRD of the gels and concentrated solutions. And we saw that the peaks that are observed from the pure stacks versus the mixed stacks still are still maintained. So there are no new peaks that are arising, and definitely, and that shows us that most likely they maintain the self-sorted structures upon assembly, co-assembly with slow acidification. So finally, to see whether this spatial temporal control in the organization of the chromophores can modulate the energy transport, looking at the photoluminescence, we can see that clearly the spectral profiles of those that are assembled with GDL versus those that are assembled with HCl are very different. So the main thing here to look at is that those that are self-sorted, we can see that the signature vibronic peaks for the donor, for the OPV3, are still very much intense and not as much quenched. But then those that are kinetically trapped into the co-assembled structures, the quaterthiophene signals are very predominant. And those with the third component, with the NDI, we can see that it's still much more quenched as compared to the ones in GDL. So I have to also mention that for the HCl assembled system, the fast system, these spectra are collected right after the addition of the hydrochloric acid. So in order to compare the different time points, I aged those samples into 20 hours, so the same time point as when I collected the gluconodelta lactone, and saw some reorganization because of the reappearance of the vibronic peaks. That's a signature of the OPV3. But then the reorganization still is more like a co-assembled system rather than a self-sorted system, because the peaks are not as intense as in the majority self-sorted system. So in this section, I highlighted two different things from the molecular engineering of optoelectronic peptides. Basically, that we can use straightforward design parameters to control the properties in the molecular, to control the different packing packing states so that we can control more macro scale properties such as the device performance. And then also we can manipulate the design of the monomers in such a way that we can control the energy transport processes. So from learning how to appropriately endow functionalities into these optoelectronic peptides in order to really understand how we can biointerface. I think this is a good switching point to describe my postdoctoral experience, where I then went and developed some biohybrid platforms and learned how to do so in order to better understand how specific nanomaterials can affect biological function. So going from understanding how to develop and design nanomaterials to understanding what type of biological models, or how can we engineer appropriate biological models in order to understand nanobio interactions. And so when people think about nano safety or nano safety by design, of course, it's associated with nanotoxicology. And a lot of people, I usually think that looking at toxicological studies, that it's a one size fits all at different length scales. But it's actually not the case, because we have to think about the nanomaterials interacting at the nanoscale with the biological components, and the effect of those interactions in whatever form that may be will be different at the cell level, at the tissue level, and at the organ level. Although they are interrelated, the impact and the types of parameters to measure will be different at different length scale. So then from an engineering perspective, it's important to understand how can we develop appropriate model systems that will allow us to measure the right parameters to understand what's happening once we expose a specific cell or tissue or organ to different doses or different lengths of exposure to nanomaterials. So in the interest of time, I will only be showing two different biohybrid systems. First would be micropattern cell pairs, which I did this work together with my undergrad mentee, Shayo Eweje. And we looked at lithographic techniques and utilized that to essentially normalize the geometry of what we call as cell pairs, which can be considered as the smallest functional unit of a continuous tissue. So in this case, we used micropatterning approaches in order to create an endothelial cell pair, because we wanted to screen the effects of different nanomaterials into into vascular barriers such as the endothelium. So by normalizing the geometry, it makes us much more easier to quantify the changes in different aspects. So morphology of the cell and the intracellular gaps between the cell pairs, which is important when we're thinking about barriers, right? And then also the localization of focal adhesion complexes, which is something to do with also the barrier integrity of these types of cells. And then using a similarity scoring index in order to much more quantitatively compare different doses and different nanomaterials for different types of parameters that we want to understand. So developing unbiased metrics around micropatterning, which allowed us to normalize the structure of these cell pairs. So on the tissue level, this is another work that I've done with my mentee, my graduate student mentee, Sangkuk An. We utilize the cardiac microphysiological platform based on muscular thin film in order to look at the tissue level effects of nanomaterial exposure. So traditionally, or the first generation of these materials, the deflection of the cantilever or the muscular thin film is what's being tracked in order to extract the data about the contractile stress. But for toxicological screening or safety screening studies, of course, at a certain extent, we want to do chronic studies and want to do that in the easiest manner possible. So in the version that I'm showing right here, essentially it's a muscular thin film that is comprised of cardiac laminar tissues on top that is instrumented with a microcracked titanium gold, so that the changes in the resistance as a function of the strain into the electrodes will allow us to much more easier track the changes in the beat rates and the twitch stress, and can be correlated with the contractile stress as well. And so the instrumentation, or the addition of the microelectrodes, is actually adapted from a design of another postdoc in the disease biophysics group, Johan Lind. So I wanted to emphasize that. But here, what we did is to present a pseudo 3D topography into the cardiac laminar tissue, more thinking about uh, having a more appropriate environment for these cardiac laminar tissues that we are testing. So the fibers are aligned, promoting alignment into the cardiac tissues, and the fibers are the ones that are presenting a 3D pseudo 3D topography. It's also important to note that these polymer fibers are a composite material between polycaprolactone, which is a spinnable material, so it can be easily fiber manufactured, but also it's in composite with polydopamine, which is a chemical analog of the bioadhesive component of the muscle foot protein. So it improves the bioadhesiveness with the cardiac laminar tissues that we are um, putting into these biohybrid platforms. So when we compare the results of the study with those that are observed in the conventional assays, we saw the same trends in the different doses of exposure, both in the test nanomaterial, in this case titanium dioxide, and also the control, which is silver nanoparticles, which is our negative control, and we know that it will have a negative effect on the beat rates and the twitch stress. So this confirm that we can use that platform, and the data that we can get from the platform have consistent results from the conventional assays, such as that just by looking at the organization of the tissue after exposure or looking at the calcium transients. I also want to note that the same or analogous type of platform, the instrumented cardiac microphysiological platform based on muscular thin film, has also been used to assess the cardiac protection by endothelial extracellular vesicles or endothelial exosomes. So I just want to quickly mention this, that this is another application of this chip-based platform. And we essentially assess the effects of these exosomes, or you can consider them as biological nanoparticles, and how it provides cardioprotective effects to laminar tissues that are seated on the platform, and how it responds to injuries such as ischemia, and even to the cycles of ischemia and reperfusion. So this is another application of that type of platform. Now, I want to end this section just by saying that there are other important platforms that we were able to develop in order to study nanobio interactions. But what I want to mention here too is that for modeling the nanobio interactions, or studying nanobio interactions, it's important to not just mimic the target tissues, but also it's important to mimic the different tissues and cells that are involved in different biodistribution routes, such as what we looked at for the looking at endothelium. And we also had studies that looked at ex vivo models of skin that were optically cleared to look at the permeation of nanoparticles at different layers of the skin. So with this, I want to transition now into what we are currently doing in the group, which is essentially looking at different optoelectronically active biomacromolecules and looking at the multi-scale integration. So we wanted to conduct a multi-scale biointerfacing and use an all organic strategy for modulating the cellular behavior, or probing tissue organ function of electrosensitive tissue. So mainly there are three different directions that we are currently pursuing: developing supramolecular biosensors, and looking into responsive bioscaffolds, and also developing self-assembled structures, so that we can manipulate excitable cells using light. Uh, but the general theme across these endeavors, research endeavors, is that we utilize biomolecular assemblies in our group and endow them with optoelectronic functionalities and utilize these functionalities in order to better understand biological responses, particularly for electrosensitive cells and tissues. Now we are currently focusing on cardiac systems. So I just want to share a little bit of what we are doing with the area of supramolecular biosensors. And my graduate students, Su Lim and Yu Yao Kwang, they're working on different variants of this monomers of this photoactive peptide polymer conjugates and looking at the structure function correlation. But I do want to show some results that was actually obtained by an undergrad in the group working specifically on the monomer that's shown in the top with the diacetylene unit that can form polymers. And what's interesting is with this type of system, she was able to see a very apparent chromism, a pH dependent chromism. So looking at these different optical states, so the samples that are shown here are basically the same as also what's represented in the absorption profile that's shown at the bottom. But I think the more important question here and the more interesting result is that it seems that there's a lot of dependence into the pathway that we take based on the pH or towards the pH variation. So, and by that I mean that if you look at when we switch the pH from the basic pH all the way back to an acidic pH, and doing so in a quick manner, kinetically trapping, the optical state or the photophysical state is the same. It's just aggregated, and it seems like it didn't have enough time to re-equilibrate into what we call as the blue state for these systems. So we are currently exploring a lot of these and also doing a lot of studies in order to perform molecular recognition using these systems. Um, and the in the other directions, we are currently exploring the responsiveness of bioscaffolds using analogous systems, and we will be exploring other peptide-based systems that will allow us to monitor biological forces in situ. And as I mentioned, also looking at how we can develop peptide-based self-assembled system that can interact with cellular membranes in order to excite electrically sensitive cells using light. So just one last thing that I want to mention about our approach is that you'll see that a lot of our efforts towards developing biomaterials is really we're taking a holistic approach, all the way from designing the molecules and really understanding how that affects the function of the soft materials that we create out of these, and considering biointerfacing in our molecular design and how we impart function to the soft materials. And we do envision that these can impact in the future how we can modulate electrosensitive tissues. So for example, pacing contractile tissues, and also providing new approaches as to how we study mechanobiology, and also new approaches towards biosensing. All right, so with that, I just wanted to thank everyone for your attention, and thank you very much again to Tom for inviting me to talk to BLI. And if you have any questions, I'd be happy to answer them. My email and our group website is also shown here. Thank you very much.
Thank you. That was great. Very, very interesting work. So many interesting possibilities, and it seems like still at a very early stage with it at so many levels as you've outlined. It's a very exciting area.
Thank you. Yeah.
Questions? So I guess I had a question. So Yeah, the field is, like I said, so interesting, exciting with possibilities, and still, I think, at a very early stage. If you were to point to the one, one best application that's been demonstrated of these sort of family of materials to date, what would that be?
So maybe not a specific application, but really on the prospects of the applications that can be envisioned for bioelectronics. I know that because you mentioned family of materials, right? So when I think about organic materials that are used for bioelectronics, I know there have been a lot of emerging applications for developing pacemakers that are that are based on organic systems. And in that sense, what has been reported nowadays is that using organic materials, because of the resorbability aspect, the biodegradability aspect, and avoiding a second surgery to remove the pacemaker. A lot of the systems that I showed here, a lot of the directions that I have shown here, we are interested in how it can be used in vitro so that we can better understand different mechanisms of diseases or toxicological effects. So right now, not we're focusing not on how it can go to the clinic, but how we can develop tools that will allow us to better understand different mechanisms in vitro.
Hmm.
Yeah.
I just see so many areas with overlap of the sorts of research that's done here. Dr. Priest does, has a whole program of light interacting with cells and using light to control different parts of cells, which was the original founding research here at the Beckman Laser Institute by Michael Burns. And then we have all sorts like OCT work and microscopy work, and just seems like there's lots of areas where there could be some fruitful collaborations.
Yeah, and I know there's a lot of also groups in at BLI that are interested in the imaging aspect. So right now, the molecular recognition aspect, I know I've mentioned that it's mostly for sensing, but if you think about for probing, and if we can have the right chromophores that will allow us to reach the near IR region for deep tissue penetration, I think that perhaps we can, with the right materials and with the right design, we can also move into that area and explore some applications for bioimaging.
Yes.
I have a question.
Go for it.
Oh, sorry, I don't know how to raise my hand on Zoom. So, I'm very interested by this sort of technology. One of the questions that always comes up when we talk to engineers about this sort of thing is the sort of the longevity of these sort of constructs and organic breakdown. Can you comment on that? And your thoughts on that going forward? Obviously, a lot of these projects are developmental. There's a lot of work to be done, but that's always going to be the sort of question that people are going to ask. You showed your lovely cardiomyocytes moving around. People are going to sort of say, well, yeah, that's great, but they'll decay. And
no, I think that's a very, very important question, not just for the scaffolds that can be degraded, but also these structures that we want to integrate it with the cell membrane and maybe can be internalized by cells later on. So as Tom said, we are at very, very early stages and we're currently focusing on the materials. But actually, one of the projects that we wanted to pursue fairly soon is understanding, well, how does these self-assembled structure perform, or how they can maintain their structures under different environments, and also if we can, if after they are non-covalently linked, if we can incorporate certain cues so that they can be um, photonically cross-linked, meaning like using actual chemical bond staplers that can be uh, light triggered in order to maintain the stability of the structures, and then also exploring if they are internal, if some of these nanostructures are internalized by the cells, are they do they go through the whole endocytic pathway, and can will the lysosome, for example, lysosomes, for example, actually traffic them all the way out, and what's this time scale of that. We are aware that these are some of the important questions that we have to answer, and we are thinking of doing certain set of experiments for that fairly soon, but unfortunately we have not done so. In general, when you mention stability, I think there's two main questions: stability just from the material standpoint, do they degrade at a certain amount of time, um, and do they lose their functionality? That's another important question. But the other question is, what does the biological system do with them? So I just cited an example of like thinking about internalizable nanostructures, mechanistically looking at what will happen to them. But also, perhaps once we've gotten into the tissue and organ level, tracing at where they are stored in the body, and that kind of stuff, the biodistribution, the typical biodistribution studies in vivo. So there's a lot that we need to do in terms of that aspect.
That's also good because there's a lot to study, right?
Yes. Yeah, yeah, yeah. But those are important questions and important studies that we have to do. So thank you for raising them.
Other questions? So I have kind of a related question. So if we look at the sort of the milieu of medical devices, and especially implantable devices that are out there, where there's some kind of material barrier between the device and the tissue. I'm wondering if there's a space where, I guess the attractiveness of the approach, you're thinking, gosh, we just like to get them right into the tissue or whatever. But might there be a bridge sort of where there's maybe a family of devices that could be implantable with some barrier, where still their organic nature provides some really distinct advantages over what's done now?
Mm-hmm. The first thing that came to mind as you were asking your question is that there's actually the advantage and the possibility of controlling the sol gel transition of these materials, meaning that it can present the advantage of having an injectable device. So thinking about the structures being in solution outside of the body, and then once it reaches, let's say inside of the body, one of the differences is that it's 37 degrees, right? So the difference in temperature, and using polymers that um, that can reach that sol gel transition in such a way that they are gel once they are delivered, after you've injected them as a solution, and then achieving that. um, I know you were probably mostly asking about um, the you mentioned the material and the biological barrier interface, and what does the organic structure present. So
Yeah, or just that, you know, that there's barrier structures now. I'm just wondering for not necessarily these materials as the barrier structure, but devices that still would go inside some existing acceptable barrier structure, but the function of these, kind of like what you just said, well, the sol gel transition, this might provide a real advantage. Yeah, so sometimes getting to the final destination, it might be, you know, like a bridge where there may be a set of devices that still are implantable and have some existing acceptable barrier material.
I see. Yeah, I think this is getting into, I know we're thinking about using organic structures as a barrier to deliver certain implantable devices to a specific site, but I think it's getting to some of the concepts that are analogous to drug delivery. And I know that there are many organic polymers that are stimuli responsive, that are used in order to carry cargo at different sites of the body. And this is something that I have not really explored much, but I think that because of um, there's a lot of synthetic versatility into the organic systems, and maybe we can learn from some of the stimuli responsive systems that's used for drug delivery in order to have packageable devices. Maybe that's something that can be done in the future, and organic materials can offer that advantage.
Yeah, sort of like, you know, there was this transition from metallic stents, drug coated, and then now biocompatible stents, and so it's yeah. Okay, last chance questions for Diggs. Okay, well, Diggs, there are no. Oh, yes, sorry. Is it Pizru?
Yes, thank you.
Hi, Pizru.
Oh, hi, Herdeline. Very interesting talk. Thank you for your great presentation. So I was just wondering if you've considered looking into using your peptide conjugate platform for as a light source for maybe like optogenetic application, because they use inorganic LED, right? So maybe your platform can be used.
Yeah, I have not thought of using the structures themselves as a light source. We do not have a structure that that has achieved maybe something like an OLED version with the peptides, and but closer to the optogenetics is actually the third thrust that I mentioned, wherein it's these structures will not be a light source, but we are hoping that it can do an analogous function as what the important optogenetic proteins are doing. So also relying on an external light source, but utilizing the structures to be photoactivated and establish transient fields that can be felt by the cells at the membrane, or rather inside the cells. There's also a lot of things that needs to be done on that, but I guess that's the direction that we are looking at. That is maybe somehow tangential to your question, but also involves the optogenetic question or the aspect that you mentioned.
Okay, thank you very much.
Yeah, thanks for your question.
You're welcome.
Any other questions for Diggs? Okay, well, thank you very much, Diggs. It was very interesting, and
Thank you very much as well. Very much enjoyed being able to see some of the faces of the people here and also giving the talk to BLI.
Okay. Thank you again. Oh, clapping. Yeah. Okay.
All right.
All right.
Thank you very
Thanks again. Bye-bye.
Bye.
Bye.